Hello everyone and welcome to Molecular Biology Online. My name is Keenan and I'll be teaching this section. So molecular biology is my favorite because it will take us beyond kind of what you've learned in typical genetics classes about, one gene equals one trait, or, one gene can't change its function over time. The best part of molecular biology and molecular genetics in this case is that we are going to see how genes are reactive things, how they serve as tools of adaptation for the organisms that they serve as blueprints for, and how dynamic DNA can actually be. So, just to get started, we can do a fun example where we see that genetics has produced species, for example, that are well adapted to their environment, and in some cases even come into conflict with each other. But as we can tell, humans can make changes to these. And one of the important things about domestication is that all the qualities to get to a beautiful Pomeranian like Scout, my dog, they all existed in a wolf. All the DNA blueprints are pretty much the same between wolves and dogs, so similar that wolves and dogs are closer related to the coding, like the literal blueprint genome, than wolves and coyotes actually are, which is amazing. And what that word is that we're going to get into eventually is epigenetics, the ability to dictate not only what genes, not what genes exist, but which ones are on and off, at what quantity they're on and off, and the timing of that on and off decision. So a little bit about me. I use basically genetics to determine how tumors are going to react to certain personalized therapies. In this case, what I did was essentially compare trials from a therapy called R2-CHOP versus a standard therapy of R-CHOP. All that really happens in this case, in the survival, we can elaborate on this or you can watch another video. Basically, if you have one of these three mutations in your tumor, in your lymphoma tumor, and you do standard therapy, you end up in this survival line, but you end up in this red survival line here if you take this augmented R2-CHOP therapy. That's something along the lines of what I do. We're going to learn how to use bioinformatics tools that can produce things like this, these, all kinds of stuff. And especially given that this is the online version of the class, the bioinformatics is something that I will emphasize and really want to stress that you learn, because this can apply to all kinds of not only things in medicine, but just using systems like this to get really good results. And we'll also examine where results shouldn't be trusted as much from bioinformatics. That's a very important point that people don't actually emphasize in a lot of these classes, so we'll talk about that. Hooray, this is a pretty good comparison of where we're going to be with Bio 388, which is the version of this class that we are doing. So technically this online version, I don't know if it's going to be called 388. Essentially it is just molecular biology of genetics. The way that I formatted this is something that it's kind of a revised version of how we would have had this class if it was fully online before the COVID pandemic happened. So in this case, there is going to be material content that I will want to assess, and it will be more applicative content. It's not going to be stuff like, what does guanine bind to, for example. I'm going to probably try and test you a little bit here, but that's just the only exam. That's the only time we're going to have that. Most of what I want you to be doing is having these periodic writing and discussion periods, and in a lot of cases, this is going to come on, through an online platform, something like a Moodle, for example. And one of the other good things is that we're going to let the primary literature drive a lot of what we find. So if you are interested in behavioral genetics, or maybe you're more interested in plant genetics, you can find journal articles and kind of talk about them and see where those applications take you. The final for this class is a written grant. So what that means is basically a research project within genomics. But again, you're going to have the freedom to explore an arena that you want to. So previous students of this class have examined behavioral genetics, physiology, cancer. It really depends on what you want to do, in your career, and if it helps, kind of looking deeper into that. Okay, so as we get, kind of rolling for today, which mainly is going to be like our genetics overview, stuff like that. This is a 300 level styled class, so any student that is viewing these or coming across this, you should be, pretty well versed in the basic ideas that we're going to address here. And that's why once we get into really high ideas like epigenetics and like how genes decide how to turn on and off based on the environment, that's where things are going to get, really exciting and new. If you are pretty well versed in genetics, a lot of this is going to be review, but it's going to be good review to kind of go over how that, how it's going to relate to the more important stuff that we're going to study. So in red, when you see a red title, this is something that's super fair game. I really need you to know that. I had a really good professor who was very good at selecting information. His way of doing it was, I would like you to know this. This is my version of that. I would like you to know this. It is very fair game. Blue is different. Blue is like, okay, it's a very good detail. It's really going to help if you know this, but it's not something I'm going to, expect an explicit, true, like full-on understanding. It can serve as a detail for something bigger, but you can kind of relax when you see blue. And green, this is where we're going to mainly talk about stuff that, hey, this is really applicative to something in maybe psychology, behavior, medicine, something cool like that. Here you can relax. You don't need to take notes. I learned that the green slides are definitely the best because this gives everybody a chance to kind of sit back and see, okay, what did we just learn and how is this connecting? So given that we can't be in a physical lab, most of these projects are going to be surrounding data that I will give you to interpret and your ability to create figures based on that or written sections that assess it. But we will have a larger bioinformatics section that will actually put you through and teach you a lot of the tools that pretty much anybody that uses bioinformatics can use. This will be centered around cancer genomics. But again, hopefully these tools represent a kind of a dive into how you can take data and interpret it. And like I said in the top, this is a very good way to show you what you should trust when you see bioinformatics data out in the wild, which it is becoming so much more common. Hooray! So you can fill one of these out if you want. It's just an intro card that helps me get to know you and kind of see like where you're coming from, things like that. You can send that just into me, and that can definitely be something that can help make the class very customized and personalized, because that's kind of my teaching philosophy, is to make sure that I'm teaching to the class. This is always a fun slide too. So like when you consider a Moodle discussion, or when me and you are discussing things, consider different perspectives. So for me, and in this case, I usually try and have students identify what is the perspective that you're best at, which is the one that you are not the best at. So for me, what I'm the best at is looking at the benefits of whatever I just saw. This is great. This is exciting. How can I teach this? That's kind of my instinct. The area that I am not very good at, and this is a detriment to me as a scientist, is cautions. Scientists have to be pessimistic. We have to see a lot of empirical evidence before we really say, okay, there is so much truth to this that we can consider it objective truth. I get too excited by stuff when I see it, and sometimes I have to check myself and make sure I am thinking about the cautions or the negatives of a study or something before, jumping in. So everybody has different talents and perspectives, but it's so good to work on where you're coming from in a discussion. Think about this as like a very good way to grow. Cool. So these are 6 types of questions that, in some cases we will be able to address, maybe not in every single case. And as you can tell, they're a lot like my domestication class questions, because that class is much more interdisciplinary, things like that. And I don't want to share too many of the questions that we'll encounter with genomics, because when we get there, you will have had to encountered previous stuff to apply it to that question, so that when we get to a big question, you have the basically ammunition to defend or justify your ideas on that question. But you can pause here, kind of take a look at how you can frame questions. The other good thing is that science is presented as this thing like, look at all these facts and textbooks we have. Science is a creative field. We want to set you loose and have you be a creator, So always be ready to start looking for questions, less so answers, when you get to certain points in your life. That's a more powerful tool to have. All right, let's do this. This is the beginning of most biology classes that I teach, is the idea that you start with this unit of life that we can all agree upon, a cell. But it is made of things, chemistry, that outside of itself and in an uncoordinated fashion is not alive. And so when we deal with viruses specifically, that's when this question, you start trending the line between alive and not alive. But in this case, this is a very good place, I think, to start, especially given that this class consists of people that are interested more in biochemistry, chemistry, alongside true biology. I shouldn't say true biology. I feel like that's exclusive. Okay. So first things first, Mendelian genetics. How does it work? Where are we going to go from here with this? And that's kind of the point of this class, is to break beyond this. So usually in class, I make some sad joke that my undergrad genetics professor did. What are the phenotypes of these 3 dogs? And he usually said dumb, dumber, and dumbest. poor labs, because they really are really smart, and I get a kind of a mixed reaction on that sometimes. I thought it was funny when he did it, but I feel like there's a lot of lab owners that were not thrilled when I say that. So again, you have these PDFs, so that's why the text is there, so that if there are certain sections of the video that, you feel comfortable on, skip it. That's why it's a video, okay? That's why this is an advanced class. But I do want to make sure everything, like even a slide like this, that, obviously we know this stuff is inherited. I've taken a 200 level genetics. I just want to be here and elaborate on stuff. The interesting thing was that this idea was not, there was not a consensus. Everybody was like, you're just a blend of your parents. That's it. But the fact is that siblings, and as we see here, all these little dog siblings, they were different. Here are the two parents. They're not just 50-50 blends each time. There was a ratio that we weren't seeing. These two hypotheses were tested, and they have not mounted enough evidence to be any good. So actually, focus on this one. A lot of people were like, parents just pass down their physical traits, that's it. Nah, if you cut the tail off a rat, its babies will have tails. And we pretty much performed that experiment. That was pretty sad. But this one we're going to examine. This is Lamarckian evolution, that organisms have basically, single organisms have the will to survive, and those that with the will to survive will survive, basically. The unfortunate example is always like, well, giraffes will just stretch their necks, and the ones that stretch the most will eat the most, done. Versus what we know now is that likely in most populations, you have variation in populations. The better variations pass on more offspring. We'll examine Lamarckian evolution later, though, because we will see that there are examples of that make you question. Okay. This is, again, something very similar. You've seen this in intro, everywhere else. Mammals, in this case, sexual organisms, they are going to pass along a varied half genome each time that will combine into a unique organism. And this is awesome because unique organisms, you've never existed before. And we'll even get into twins that have the same genome. We'll see how they're different. And that's where, again, the point that I brought up in the beginning of these slides is the genome is a developing thing. Just because you're born with that blueprint doesn't mean that there are changes on the way that not only you can make that, or that the environment will make on you. Okay, this is, again, pretty just detailed slide showing that in a given, in a typical cell division, you're going to make two daughter cells. They're identicals. You want that. You don't want variation as your body is growing and, the systems of your cells. You want that consistent, okay? With meiosis, you want variation because varied offspring means that in a traumatic environment, you'll probably at least produce one that works. That's the idea behind varied life. It's not to mess things up. It's that the environment is an aggressive place to be. And you want offspring where at least one of them is going to make it. That's the idea behind unique offspring. So, that, basically chromosomes cross over like you see here. They exchange pieces of their sister chromosomes, essentially, from the mom and the dad pair. And you can make unique combinations, and everybody has this happen. So, at this point, we know sexual division, things like that. Okay, again, this is where in the PDF you can go and, renew yourself if you need. But what Mendel figured out was these populations had a ratio. Okay, they were not hybridizing. They were not 50-50 blends. If it was a 50-50 blend here, you'd expect like a pink, right? No. And when he figured it out that if you could create heterozygous flowers, ones with a version of the gene that was purple and a version of the gene that would codify for white, they would come into a 3-to-1 ratio. And again, remember, what Mendel's working with here, people literally have no idea. We know at this point that parents pass something on that make offspring look like them, but we didn't know mathematically why. Mendel's a weird dude, too. You should read about him. He talked to the pea plants. He was super weird. So, phenotype, again, you've seen this. Phenotype is the trait that we see or we observe or we measure. Genotype is the alleles, the versions of the genes that are actually creating the phenotype in this case. This is where the ratios change. With genotypes, you can have double dominance that can codify for, a purple dominant phenotype. Heterozygous ones where you at least have one copy of the dominant gene, they're both going to, create purple flowers in this case, because just one copy is enough to create this phenotype. We'll see basically, and this is an advanced class, the reason dominant genes are dominant is that they're a working version of the gene. This capital P is a working version of the purple gene. The lowercase p's, these are non-working versions. That's what recessive alleles essentially are. They're non-working versions. In this case, there's no purple gene here. That's it. And that's why, that's how you can tell that something is recessive or dominant, is that essentially these are recessive because they don't work. That's why you get this white phenotype right here. Okay, alleles. These are versions of genes. So pretty much you have, humans, for example, even have the same genome for the most part, right? Some of our genes between different people have different variants. So just a small change in there that can basically create a gene that is capable of producing the purple pigment, or, for example, a gene that is incapable of producing that purple pigment. So you can visualize it like, oh, this is the white allele, this is the purple allele if you want. Or like I said, you can look at it more from the mechanistic view that we're going to get into in this advanced class, which is these recessive ones, they just don't work. This gene is defunct, right? Okay. Now, how genes pass on, again, it's all dependent on meiosis. When you have a typical diploid cell, like humans are made out of, during the sexual meiosis process, each of these chromosomes that contains each of these alleles, it's going to split randomly. And this is totally random which way this split happens. And that's kind of going to contribute into the randomness of sexual organisms and their offspring. So here, these are more details on recessive and dominant alleles here too. These are really good, and that's why this is in red. So you'll notice most of the reviews in red because that's definitely an expectation that you should be coming into this class with. If you don't have that, if you see something that is unfamiliar though, that's the chance to reach out to me during the first discussion sessions, which you should be coming with, any specific questions and just bigger, exploratory questions if you do feel comfortable and ahead of things. So that's good. Okay, so given this 50-50 ratio of how you pass stuff on, so see, it's going to go that way, it's going to go that way from the male organism, and I would draw just like that. So right now what this Punnett square is doing is looking at the possibilities for one gene. In this case, you're going to have heterozygote, double dominant here, since capital T. Then over here, oh, that's going to, there we go. Then you can have recessives. And then in each of these, you're going to have the heterozygote, where you have the capital T and then the small, right? So this is just a way to visualize what your possible offspring are going to look like in terms of this gene. Now again, like I said, review, because we are going to break this concept much, much more when we get into, the real way that a lot more of your genes actually behave. So do remember that Mendelian genes, this is not the whole story. Okay, so we've kind of talked about this. This is just another great visualization of how, what kind of phenotype are you going to produce, and, essentially how we can predict that. And how we predict that is meiosis. Okay, so let's get into some medicine. Take a look here. These are two parents right here. One is a carrier of the cystic fibrosis gene right here, which means that one of these genes that serves as basically a functional cell transporter, a cell membrane transporter that basically funnels mucus, or, well, at least I think it actually funnels one of the elements like chlorine or something, and that causes mucus to go away, or at least be more regulated. If you're born with two defunct copies of this, mucus builds up in this case. Okay, so again, this is how a recessive trait can pass on. This is where, if you're a genetic counselor, this is where you come in with this family and say, okay, here are your odds. Here are your odds of a potential child not only carrying this disease, but having a normal set of lungs in this case, or a child that carries the disease. Okay, so again, one dominant copy is enough because then you have one working version of the gene to make the cell transporter that this gene is coding for, the cystic fibrosis gene itself, that will funnel out the cell, the cell equilibrium and make sure that that is a, something functional. Okay. Oh, I should have gone to this slide next. Okay, here's CF at the functional level. Okay, here's an individual with heterozygote or double dominant. You have this nice, yep, it was chloride. You have this nice working transporter. This channel is going to flow out nicely. If you have the mutation, though, this channel does not work, and you cannot come to equilibrium as you would like to, and mucus will build up, and this is where you get the disease physiology. Okay. This is just another detailed view, and this is a detailed class. So what this is, in cystic fibrosis, is you're on chromosome 7 where this gene is, CFTR right here. The mutant allele is a deletion of these 3 nucleotides, which means that you are never going to get this phenylalanine here, which means at this stage, in this regulatory domain, it basically bottles things up. So by taking out that single amino acid, you basically ruin the function of this transporter. So that's why one working copy still means you have a working good transporter in all your cells. It's not 50-50 at all. Okay, let's go to a dominant disease trait. This is Huntington's. If you have one of the copies in this case, that is enough for disease. One single copy of the dominant gene means that you're going to have the condition. So now the table's been switched. Now a dominant gene is causing the condition. What this means is that the recessive gene, in this case, probably functions normally. So this is where you have to kind of recharacterize things. With a dominant disease phenotype, this is where you have something that is amplifying its effect, or it is causing a dysregulatory effect of the normal gene. It's not necessarily deleted in this case. You've actually probably added a function that you should not have. And in this case of Huntington's, you've added the ability, unfortunately, for this protein that this gene produces to cause the buildup of other proteins, which is detrimental to the brain. Let's take a look at this physiology. Okay, so this glutamic acid. Nope, wait. Glutamine. Dang it. I have to look at that. In my first video, I ruined one of the amino acids. Essentially what happens, what this trait is, what this allele is too many of these repeats. This can happen a number of ways. We'll see how insertions can be made, and so how this allele came about once, essentially. But this mutant sequence is essentially that mutant allele. This is a dominant allele. Yeah, it was glutamine, dang it. When you have too many of these repeats, it becomes a dominant version of that gene that only one is enough to ruin the entire thing. Because if you have one copy running around inside the brain, it's going to cause all kinds of pockets of these repeated proteins to come through. And that's what will cause disease and basically plug the brain. Okay, so this is a very good question that is pretty tough in the first time you encounter it. Why don't dominant traits take over, right? They're dominant. They shield the recessives. The big reasons that recessives have a huge advantage is that they hide in carriers. Okay, these are individuals that don't express the recessive phenotype, but they can carry it. They can only get selected against fully recessive individuals, okay? And now when they're good too, when recessive alleles are good, they're 100%. You know, they are fully, they're fully being expressed. If you have a good dominant allele, that means that heterozygotes can be passing that on, but also 50% of their offspring won't get the dominant allele, right? So in this case, when you're fully recessive, all your offspring are going to get a full dose of your recessive allele in that case. So this is kind of exemplified on the bad end through inbreeding. So inbreeding is negative because you essentially have a family, and this is true in almost any individual. A lot of us have a lot of recessive alleles that only exist as one. We have a dominant allele that makes it okay. The problem is that even though that passes along in a family, even if it's rare outside, if individuals breed within their family, you're much likely to come across a rare allele with a rare allele and produce a double rare allele offspring. This is why inbreeding usually has a deleterious effect on offspring. We see this in dogs, a lot of agriculture animals that we force to inbreed. you see that the rare bad gene variants that normally would never show up in the wild with random mating will show up then. The strange thing is that, and this is why you have breeds of dogs that don't have inbreeding issues, for example, is that eventually you can basically purge out these genes through multiple inbreeding rounds. And this is basically how you create dog breeds, to say the least, or any other breed of domestic animal that, with these two right here, you will end up with a lot of these individuals, and they will not survive in this case. But you will end up with other individuals that are double dominant, for example, and essentially those ones eventually mathematically win out if you keep inbreeding. But again, remember, this is a very human thing that we're doing. Okay, so that's Mendel. And Mendel is very important to understand. But do realize that genes behave different ways, and we're gonna go in this class and see why genes do what they do, and that's kind of what has been the intro so far. Look at incomplete dominance in this case too. So if you have a parent that is double white and a parent that is double red, they pass on one gamete each. You're going to end up with pink. Oops, there we go. What that means and what you're actually seeing in this organism is that every other cell is like, there are 50% red cells and there are 50% white cells in this case. So what that does is that to our human eyes, it looks like a blend, when in fact, if you look at a microscope, it's one red, red, white, red, red, white, red. During development, basically the embryonic cells compete with each other, and 50% of them will ultimately express one of the genes, 50% will go to the other. What this means is both of these are dominant genes, but only one can be active in a single cell. So in that case, your heterozygotes are going to be essentially blends in that case. But physiology-wise, it's random. Okay. So, epistasis. This is where one gene is controlled by another one. This is your introduction essentially into epigenetics. So if you want to pause the YouTube here, you can kind of take a look at this big board. But what I would do is first just ignore the E gene. Ignore this big E, okay? Let's focus just on the B set of alleles. So the capital B and the small b, right? These are black and brown labs. The brown lab color is recessive. Most Labradors, their dominant gene is black. That's the melanin gene in Labradors. Double dominants are going to be black labs. Heterozygotes are going to be black labs. And brown chocolate labs are actually just both recessive versions. So where do you get yellow labs? Yellow labs are the E gene. Okay, that's what this is. The E gene essentially, and it's called E for epistatic master gene. The E gene in this case is totally different than the melanin black gene or brown gene. It's the controller, it's basically the on switch for the brown or the black gene. So if it is double recessive, the black or the brown gene doesn't matter, because it's the master regulator. So in a yellow lab, you may have the genes for melanin that are ready to go, but if you're double recessive, which remember means like you're not working, these genes aren't working really, if you're double recessive for the gene that controls all melanin expression, you're going to end up with a yellow lab because you're never going to express any of the black or the brown alleles in this case. They just don't make it to expression. So that's how this is how genes start interacting and why you have to pay attention. Okay, codominance. So we looked at incomplete dominance. That's where cells basically, like at the development stage, they fight over, if they're heterozygote, 50-50, who gets, which expression. Codominance is, a lot of the time, you have dominant versions of genes, and both of them can coexist on the same cell. So in this case, this is blood type is a great example here. So remember, genes are coding for surface proteins. Well, genes are coding for proteins, sorry. In the case of blood genes, you can actually see it. Their destination is on the surface. You have different versions of the blood gene. One that produces no receptor. It's a recessive allele. You have two different dominant alleles, which means you have, different proteins that express. So both of these genes can express, and they have to kind of share the space if they are both expressing in someone. With I, it means that you don't have that receptor gene. So in this case, if you have 2 copies of the A gene, yeah, you're going to express just A. If you have 1 copy of the A gene, it's dominant. You're still going to just express A. Same thing for B. But remember, this is just a different version of the same gene, but there's 2 different dominant versions in this case. And this is where it gets special is, if you have neither, then nothing expresses here. But if you have both, this happens in all your cells, not the 50-50 of incomplete dominance. If you're AB positive blood, then that means you have both receptors on your blood. So when you get into medicine, what this means is that you cannot give this blood, which is full of what we call actually antigens, to other people. You can't give this AB blood to a person that has O, because their immune system does not recognize these as self. They don't recognize them as part of the body. They think that's pathogen invading the body, and that will trigger the immune response that kills not only these cells, but the rest of the body of the recipient. So that's not good. That's why O blood is so desired when it comes to donating, because they don't have any of these antigens. O blood can go to anybody's type of blood, A antigens or B antigens, or both. There's a lot of drawing on this slide, so we're going to talk about this because this is a great medicine example. All right, almost done, right? So polygenic traits. These are also called quantitative characteristics. This is where lots of genes determine a trait. Height, for example, is a trait. It is determined by 100 to 500 suspected genes that contribute to height. Usually, mathematically, the more of these gene variants you have that are the height variant, the higher, the taller you get. It's not a single Mendelian thing. And in a lot of cases, some of these genes actually interact and kind of augment each other. So it gets even more complicated than that. So the biggest thing to understand of what we'll try and cover in this class is that most diseases, most traits, most everything is you're depending on a lot of genes. It is pretty rare that one gene equals something, okay? And this note right here kind of highlights that, is that when we sequenced the human genome, we thought there was going to be an Alzheimer's gene. We thought there was going to be an obesity gene, a diabetes gene. It turns out that genes interact in these massive systems within cells. We are understanding that better, but the one gene, one trait thing, that doesn't exist. Everything plays a role, and these genes are going against or with each other in all kinds of movements, and that's the point of this class, is to understand that. This is a very good developing idea. Okay, so just what I said, monogenic traits, one gene, one trait. Polygenic traits, just like what we talked about. And this is something with eye color that's a very good example. There are different genes that contribute to the blue eye color. There are some that are more powerful than others, but ultimately it's a collection of genes. And this is the idea of omnigenetic traits, those that are determined by massive numbers of interactions throughout the genome, and that throughout at least the development of an omnigenic trait, eventually all genes are somewhat involved in that path. This is a bit of a stretch when it comes to how many genes can interact, but it is definitely gaining ground as an idea that can really help us understand the fundamentals of genetics. Okay, this is where the developing genome idea starts picking up some steam. These are complex traits. What we've been talking about are complex traits, things that exist on a spectrum. And not only do they get determined by genes, they get determined by the environment, definitely, okay? Down here are some examples. So if I love the example of risk-taking. So imagine these are the number of individuals. So within this section right here are about 66% of individuals lie within this spectrum, okay? So let's consider the squirrels that come on campus, for example. If we're looking at risk-taking, here's the population of squirrels that exhibit no risk-taking. They are extremely fearful of humans, for example. They will never come to campus. They will never come and interact with trash cans. And thus, they won't get as many rewards from that. They also won't be in an environment with humans around that actually deter a lot of their predators, right? So over here are the squirrels you see on campus. These ones have massive risk-taking capabilities. They have low fear genes that determine their neurological fear basis. They reap the rewards of being on campus, which is trash, protection from predators, things like that. These are also the same squirrels that have really high risk taking, that probably take a few too many risks, even in a safe environment like a campus, and that's why they get selected against. So when you look at the spectrum of populations, consider the extremes on each side and how genes are playing a role in that, but also how the environment basically triggers certain genes, because that's what we'll kind of talk about too. Immune strength, another great one, especially right now. People have really hyperactive immune systems. Some people have low active immune systems. Most of us exist here. People on either side of the extremes have different challenges that come with that, though. Okay, so we can break the single gene theory another way, where one gene can control multiple traits. In this case, this is a very good example of how things were domesticated. In a lot of cases, domestication comes along with many different kinds of traits. So in this case, not only is Scout, my dog, very different from a grumpy wolf. Scout is smiley. Her ears flop. Her tail is curled, for example. And you see these traits in a lot of different dogs, even the ancient breeds that were initially domesticated. You still have some similarities, as you can see. Apex predator, Scout, good girl. So how do you change one gene and change entire organisms? If that gene controls a massive pathway, like glucocorticoid receptors do for fear and stress, if you remove these receptors, which is essentially what you do when you breed animals to be less fearful, if you get rid of this gene all of a sudden, a lot of these glucocorticoid receptors, you don't have as much of the response, which is a cellular response of fear, right? That's how we've basically made dogs into these non-fear animals, at least non-fearing to us, right? Interestingly, this pathway has a lot of different cellular activity during development. And by development, I mean like embryonic development. That's why in a lot of cases you see embryonic development differences in ears, skeletal structure that dogs have between wolves. So that's when one gene, such as a receptor for glucocorticoids, it comes as a package deal. It's that big of a gene, for example. So what this is essentially saying is that not all genes carry the same weight. Some are more powerful than others when it comes to the number of traits that you see if you change them. Okay. This is going to start getting into our bigger points too. Environment can alter phenotype. Okay? Phenotype is not something that is strictly only determined by genetics. In this case, temperature can change how this plant looks. And do not think that humans are below this idea, right? Humans and mammals are highly determined by an environment. A simple example is stress. There are genes inside of each one of us that will likely lie dormant forever. They will be inactive. They are genes that likely served some role in the past. I don't know why it's cut off. This is another example of something that my genetics professor once told me. Genes are the bullet in a gun. Environment is the trigger. If you put an organism up against chronic stress, you will activate specific genes, specifically neurological genes, for example, that will condition that organism to react and to behave certain ways because it's a survival mechanism, okay? Stress will activate genes, and it usually does in a chronic setting in a lot of cases, sadly. So acute environmental interaction can sometimes fix patterns too, including human insanity. So this story is about a Russian author called Dostoevsky. He was lined up in Siberia as a prisoner, and he was set to be executed, and the punishment that they actually got was a fake firing squad. So all his friends are lined up. These are all blanks. They fire, but one of his friends, he points out, who knows which one it was, the stress of that fake death right there, that firing squad that didn't kill him, he was insane after that. It was gone. The changes that went inside his cells and his brain were permanent. They never unfixed. Humans are complex, but we're not immune to what the environment can do to us. Okay. This is very true in dogs as well, where chronic amounts of stress and a traumatic life can cause, terrible, terrible conditions, and basically, you can see the body reacting in this case, in some cases, and sometimes that is fixed patterns of abuse or stress, but you can fix that. You can come back from that. You can turn off those genes that this animal's experiencing. Not every time though, and that's the sad thing. That's why sometimes you get animals that are they're too far beyond what we can do to help them, because in a lot of cases genes fix. They don't come back. They stay on forever, for example. Okay, so this slide is kind of getting into some details about what, we didn't know why for a long time certain genes expressed and why they didn't. And importantly, expressivity is how high a gene turns on. So in this case, what we're going to see in all four of these cases is the mutant phenotype. Imagine all these people, in each case, they all have the mutant setup for the gene, right? The mutant phenotype. If you have complete penetrance and narrow expressivity, it's either on or it's off, and it gets all the way through. Penetrance is essentially, do you actually see the phenotype if you have the genotype? In this case, yep. So with incomplete penetrance but narrow expressivity, you still have it basically an on and off, basically black and white like you see here. But even though all these people have the same genotype, some of them don't actually end up expressing the phenotype at all. Strange, but again, that's where the environment usually comes in. Here's where complete penetrance, so everybody's expressing the phenotype, but not to the same degree. Broad expressivity means that the dosage of a gene can be low or it can be high, right? And we're going to examine this concept quite a bit, that genes can come in different quantities, and that's going to basically change the kind of trait that you're going to have. And again, this is the final one. Sometimes the gene, the phenotype just won't come through, and it's also being broadly expressed where it does come through. But expressivity is the most important thing here. Basically that some traits and some genes, the quantity of how much they express is very determinant about what you see from them, okay? This starts to get us into the nature-nurture idea. Twins give us a very good idea of this, right? So in this case, we have identical twins in white in the bar graph, and in gray, fraternal twins. Fraternal twins share the same environment, remember. So do identical twins, but identical twins share the same genome. So when we look at something like height, look at this big split between identical and non-identicals, right? If you're an identical twin, pretty much 90% of the time, you have the same height as your identical twin. So that means there's a greater genetic influence, especially when compared to the, let's just delete these, especially when compared to the fraternal twins, which are basically just regular siblings, born at the same time, same environment though. So as you look at all these, you see that there is a genetic predisposition towards these identical twins. See how many more identical twins share the trait in each case here. That showcases that there's a genetic influence. And interestingly, it showcases how much genetic influence actually exists in behavior, the things I've highlighted here. You start to get into things that have greater environmental influence. And that's where we can go into these 100% trait types. Blood type, that's a great example of, high penetrance, binary expressivity. You either have the blood type or you don't. And if you have the genetic version of the blood type that's identical, twins have the exact same one, exact same A or B, that's it. Something that is 100% environmental, though, is going to be something like language, and not necessarily the human capacity to learn language. I'm talking about splitting two identical twins and teaching one German and one French, right? That doesn't matter on genetics at all. So by the end of this class, you're going to be able to, this shouldn't be in red, this should be in green, sorry. By the end of this class, you should be able to look at this picture and understand multiple things that will determine what an organism is. And not just what an organism is, like this is a human, who that human is. You will get into personality. We will examine, how much genetics determines that and why that's a dangerous question, but a very exciting question too, but one that I want you to examine very critically as we get into this. Okay. I think that's about it. This is just a good review of the central dogma of genetics. Remember, we've been talking about DNA this whole time. DNA has to express and copy its codes that you have stored safely in here to an RNA, This RNA is going to leave the safety of the nucleus, and it's going to be translated into the language of amino acids. Because remember, here's nucleic acids, and here's amino acids that make proteins, polypeptides, and we'll cover this, but remember that proteins are what cause these traits. Your DNA doesn't go and act and like create melanin. The melanin genes that act and like showcase like we saw in the dogs, they are proteins. That's what's causing something in a species. DNA has very few functional roles. We will get to highlight them in this class, but it's proteins that do the work. Okay. This is kind of one of our last ones. So when we talk about mammalian X and Y traits, this is the breakdown that you have to keep track of. Remember, mammalian females are going to have 2 X chromosomes. Mammalian males are going to have this Y right here. Now, the Y is an ancient ancestor of the X, but it has been so degraded and left behind, basically, regions of the Y throughout history, and I mean million year history in mammals. You lose like regions, but as long as you have a single X, you're going to be okay because all the genes that you need to function are still on an X. No organism can function as a YY. There aren't enough genes. You have too many genes that are important on an X. The Y genes that do exist include SRY, which is the male type, basically like triggering gene. You can look into that, and we'll cover it a little bit. And a lot of, testosterone type stuff, things that start differentiating the mammal male and female phenotypes. And it will be pertinent to remember the Y chromosome because we will get into this when we get into human behavioral genetics. Okay, here's a good example of disease though that is an XY type thing. If you have a father, remember that these chromosomes are going to split, right? You're not going to pass along both. The Y is going to be in one sperm that's going to show up, and the X is going to be in the other sperm. In the 2 eggs from the female mother, from the female, this is where you can actually have different alleles of the same gene in this case. So in this case for hemophilia, if you have one of the disease variants right here, then you're going to have to split it just like a normal thing. So the only thing that changes in Punnett squares for these is that the Y is essentially just a non-genetic entity. You just don't worry about it because it doesn't have any genes, essentially, and we'll cover that. So that means that sons, though, males only have one shot at a working X chromosome gene. If you have a baldness gene or you have a hemophilia gene, that's it. That's the only copy you have. It doesn't matter. Females or daughters with a recessive hemophilia gene, for example, or a baldness gene or something like that, as long as they have a working copy, they're going to be fine. It's males. That's why you see way more males with X-related genetic disorders, is that males only have one shot at getting it right. Females have 2. Okay, so we're going to start covering ancestry and a couple other things in genomics in the next class. So should be all good.